Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, let's unpack this. Imagine your immune system, right? It's this highly sophisticated army trained meticulously to protect your body, but also, and this is really crucial, knowing what not to attack. We typically think of this whole self-tolerance thing as, super precise, tightly regulated. But what if part of this vital training actually involves embracing a bit of, well, genetic static? Like, what if your body actively turns up the genetic noise to teach tolerance?
That's precisely it. That's what this really groundbreaking new research from Nature is proposing. For a long time, we've understood specific genetic programs, the REU gene being the famous one, are vital for guiding T cells, those immune cells, to recognize your own body parts, self-antigens. But this paper, Thymic epithelial cells amplify epigenetic noise to promote immune tolerance, it introduces a really counterintuitive twist. It says specialized cells in your thymus actively increase the fluctuations, the randomness, in how their DNA is packaged, what they call epigenetic noise. And remarkably, this whole mechanism seems to operate largely independently of ERI.
Whoa. Okay, so the really mind-bending takeaway here, right from the start, is that some of this randomness we see in gene activity isn't just background clutter. It's actually an engineered system, a critical one, that stops your body attacking itself.
Exactly. We're talking about a fundamental shift in understanding immune tolerance, and it's rooted in, well, the physics of DNA packaging. It's not just a side note, it seems to be central.
Okay, physics even. Wow.
No. So, just to set the stage quickly, the thymus, that's your body's T-cell school. It's where these developing T-cells learn friend from foe, and the teachers are these thymic epithelial cells, M-times. Their job is to show this huge library of self-antigens. This paper really digs into the molecular nuts and bolts of how MTEX get such incredible diversity in what they show. And yeah, it strongly suggests this noise isn't just a byproduct. It's something they actively use, a feature.
Right, an exploited feature. And here's where it gets really interesting for you listening. This research doesn't just uncover a new, airy-independent path for tolerance. It also links it to P53, the famous tumor suppressor, and even to cancer. So we're going to guide you through how they piece this all together, figure by figure. Let's see how they unraveled this puzzle. Okay, let's start our deep dive then, beginning with figure 1.
Okay.
This really lays the groundwork, right? This idea of epigenetic noise driving gene expression where you don't expect it, ectopic expression.
Exactly. The researchers were studying these MTEX very closely, and they noticed something, well, intriguing. The overall openness of the DNA, what we call chromatin accessibility, basically how easy it is to read genes, wasn't wildly different when these specific tissue genes, the ATSGs, were turned on. But something else was shifting subtly.
Okay, so not the main openness measure. What was it then?
They looked at single-cell ATAC-seq data, which is a powerful technique. It maps where DNA is loosely packed accessible, right down to single cells. And they found the differences weren't just in the main gene areas, the peaks, but in the sort of noise outside those peaks.
Outside the peaks. So like in the regions we normally think of as closed off.
That's right. They actually define these as out-of-peak signals, OOP signals. Think of your DNA like a library. We usually focus on the books checked out the open, accessible regions, the within peak or WIP signals. But this study found the fuzziness around the edges, the slight disarray. That actually became the clue. These OOP fragments, bits of DNA in supposedly closed regions, they surprisingly predicted which rare extra genes might flicker on.
That's fascinating. So they didn't just filter this noise out like maybe usually happens. What made them think it was important?
That's a great point. They noticed these OOP fragments had longer nucleosomal lengths. Nucleosomes are the spools DNA wraps around. Longer lengths here meant normally tight regions were becoming more labile, unstable, more likely to unspool briefly.
Okay, more unstable.
And get this, they ran a logistic regression analysis, a statistical tool. It showed that the variation in these OOP fragments was a massive 140-fold more reliable predictor of a gene flickering on than the usual within-peak signals.
140 times, that's That's incredible. So much predictive power in the noise.
Exactly. It really highlights it. So it's not just about DNA being fully open there.
Right. It's more subtle.
It's about the fluctuations in the background,
Mm.
a kind of statistical temperature, you could say. It makes these dense regions just permeable enough for these sporadic, noisy gene expressions to happen.
Like a little temporary leak allowing transcription.
Precisely. And what's more, they found that if one of these ITSGs flickers on, it increases the chance of a neighbor within about 100,000 base pairs also turning on. It suggests a sort of localized destabilization, a ripple effect.
Wow, okay, this fundamentally changes the picture. It's not just on-off switches. We need to consider this dynamic fuzziness that allows this crucial diversity. It's actually quite elegant. All right, moving on to figure 2. This is where the plot really thickens, isn't it? Revealing that this noise mechanism is distinct from air and brings in, well, a very surprising player, P53.
Yeah, figure 2 is key. We knew air was critical for what's called promiscuous gene expression in MTEX. That's their unique ability to express genes from all sorts of other tissues. Liver, skin, you name it. It lets T cells learn to tolerate the whole body.
Right, the promiscuous expression.
So the authors saw this pattern. As MTEX matured, the within-peak fraction, the WIP signal, decreased. And at the same time, these nucleosomal fragments linked to noise increased. This mirrored the rise in those diverse ITSG expressions. But crucially, they found this happened even without air.
Even without air?
Yes. Even in air-deficient MTEX, this chromatin accessibility noise was present, meaning this is an air-independent pathway, completely separate.
Amazing. So the body has another trick up its sleeve for tolerance, but if air isn't driving it, what is? How did they find P53?
Well, they started looking for transcription factors or signals that might be involved, and they found a significant depletion of binding sites for P53 in the mature MTEX. Now, P53, as you know, is the famous guardian of the genome, a tumor suppressor. Its job is usually to maintain stability, prevent cancer. So finding its influence reduced suggests P53 activity is actually being actively repressed as MTEX mature.
That is really counterintuitive. You're saying the cell is suppressing its main stability guard. Why would it do that? It sounds risky.
It does sound risky. And that's what makes it so interesting. They found that MTEX actively induced molecules that shut down P53. Things like MDM2, CoP1, and SRT1. These basically tag P53 for destruction or switch it off. It's almost like the MTEX are telling P53, okay, look the other way for a bit. We need some flexibility here. We're creating noise.
We need to make a bit of a mess for a good cause, huh?
Something like that. But as we'll see, the balance is key. It's not complete chaos.
Right, there have to be checks. So let's explore what happens when this repression doesn't happen. What are the consequences? That brings us to figure 3, right?
Exactly. To test this, they created mice where P53 activity was actually boosted in the MTEX. They called them P53 cyper mice. And just as you'd predict from the hypothesis, they saw a significant increase in chromatin stability. The DNA became stiffer, less noisy.
The opposite of normal maturation.
Precisely. And this stiffer chromatin led to a drastic reduction in the diversity and the amount of that ectopic gene expression. So turning P53 up acted like a strong brake on this flexibility. It shut down the noise.
So preventing the noise shuts down the diversity of self-antigens. What did that mean functionally for the MTEX job?
Well, the functional impact was clear. These P53 cyper MTEX, the ones with less noise, they had a much lower ability to differentiate into those various mimetic cell types, where they mimic gut cells or skin cells as part of teaching tolerance. Their plasticity was really limited.
Okay, so P53 is acting like a molecular lever here. It maintains cell identity, stops them getting too weird, but you need to dial it down to get the necessary plasticity for tolerance training.
That's a great way to put it. Dialing down P53 allows the plasticity needed for diverse antigen presentation.
Now let's get into figure 4 and beyond. How does P53 actually exert this control? And what starts the noise in the first place? And ultimately, what's the big biological consequence?
Okay, so how P53 controls this is also quite subtle. They found it doesn't seem to directly bind to these noisy regions to shut them down. Instead, when P53 is hyperactive, like in those special mice, it seems to indirectly trigger apoptosis programmed cell death, but specifically in those MTEX that are showing too much plasticity.
So it's a quality control, not stopping the noise directly, but cleaning up cells that get maybe too noisy or unstable.
Exactly. It's like a surveillance system. If a cell gets too far out of line, too plastic, P53 ensures it's removed. This selects for MTEX with just the right amount of controlled instability.
That makes sense for maintaining balance. Okay, but what kicks off the noise? What's the initial spark that creates this instability that P53 then monitors?
This is really elegant. They found a strong link to the DNA sequence itself. The regions that showed this heightened noise, this increased accessibility fluctuation, they were selectively enriched for long stretches of just adenine and thymine bases. AT tracks, poly-D-ADT tracks.
AT tracks, I remember those are kind of structurally weird, right? They don't pack well.
Exactly. These AT-rich regions are inherently resistant to being tightly wrapped around nucleosomes. They naturally destabilize the packaging. This creates soft spots in the genome, making the DNA more prone to temporarily unwrapping, more noisy.
So the potential for noise is literally encoded in the genome sequence.
Yes, and this unwrapping allows for that random spurious transcription initiation. RNA polymerase can sometimes land there by chance, but that kind of activity in unstable regions can cause DNA breaks. And DNA breaks are a primary trigger for p53 activation.
Ah, okay. It's a beautiful feedback loop. The DNA sequence itself creates the potential for noise via AT tracks. That noise allows for antigen diversity. But the process can cause DNA breaks, which activates p53, which then acts as the quality control, removing cells that break too much or get too plastic.
You've got it. It's a self-regulating system. The genome encodes the potential for noise. The noise generation process itself provides the signal, DNA breaks, for the regulator p53 to keep it in check.
That is genuinely elegant. So what's the ultimate consequence then if the system fails? If p53 is too active, for instance, tying it all back to immunity in Figure 5?
This is the crucial payoff finding. Augmenting p53 activity in MTEX, remember those p53 cyper mice? It caused multi-organ autoimmunity.
Autoimmunity, so the immune system started attacking the body.
Yes, severely. These mice developed increased numbers of activated T cells and serious immune cell infiltration into multiple organs, salivary glands, lungs, kidneys, pancreas. Classic signs of autoimmunity.
Wow. So dialing down p53 and allowing this managed epigenetic noise is absolutely essential for self-tolerance. Without it, the immune system literally attacks itself.
It's essential. That carefully managed noise is critical for teaching the immune system correctly. Too much stability leads to autoimmunity.
And just when you think it's all about immunity, they connect it to cancer too. How does that fit in?
Right. They extended this principle. They looked at lung adenocarcinoma cells, and they found that p53 also regulates this chromatin accessibility noise and cellular plasticity in cancer cells. But here, p53's role is beneficial by suppressing excessive noise and plasticity. It actually inhibits the cancer cell's ability to transform and progress.
So the same mechanism has different outcomes depending on the context. In the thymus, you need to dampen p53 to allow controlled plasticity for tolerance. In cancer, active p53 limits plasticity, which is good because it hinders tumor progression.
Exactly. It's about context and balance. Controlled noise for tolerance, suppressing excessive noise to fight cancer.
So wrapping this up, what does this all really mean? This deep dive has uncovered, well, a really innovative concept. The idea that this background chromatin noise isn't just biological static, but a purposeful mechanism for immune tolerance. That's a huge conceptual leap, isn't it?
It absolutely is. It moves us beyond thinking just about specific factors like air and forces us to consider the biophysical properties of chromatin itself as a key player.
A biophysical dial, you called it earlier?
Yeah, I think that captures it. This study connects the dots beautifully from specific DNA sequences, these AT tracks, to an upstream regulator, p53, and all the way to functional outcomes like autoimmunity or even cancer progression. It's a very coherent, robust mechanism supported by so much converging evidence, genomic, genetic, phenotypic. It's really compelling.
It really makes you reconsider all the other noise we might filter out or ignore in biology. Could other random seeming fluctuations actually be doing critical work in our cells?
That's the big question it raises, isn't it? This work really pushes the frontiers of immunoepigenetics. It suggests we need to look much closer at what we previously dismissed as just background fuzz.
The potential implications seem massive, too.
Oh, definitely. Imagine if we could therapeutically tune this chromatin stability. Maybe we could tighten it in cancer cells, make them less plastic, easier to target, or perhaps loosen it carefully in autoimmune conditions to help the immune system maybe relearn tolerance.
Fine-tuning the noise.
Exactly. The potential for manipulating these epigenetic levers, managing this noise therapeutically, it's vast. Lots more research needed, of course, but the possibilities are exciting.
Absolutely fascinating. This deep dive really shows that sometimes the most important lessons aren't learned in perfect silence, but in that kind of delightful hum of genetic static. What a journey through this paper. Until next time, keep exploring, keep questioning, and maybe keep listening for those surprising insights hidden in the noise.